All right, let's dive into this explainer and zoom right into life's ultimate microscopic building block, the cell. Crazy enough, life probably didn't start in a calm pond, but in boiling hot springs billions of years ago. In extreme places like India's Puga Valley, heat-loving bacteria actually forged the very first protective cell membranes. Since these blocks are impossibly small, it begs the question, how did we even uncover this invisible world? Well, our eyes have a hard limit. Anything closer than zero point 0.1 millimeters just blurs together. That all changed in 1665, when Robert Hooke used a light microscope to officially discover cells. Today, electron microscopes blast past that, revealing mind-bending, nanometer-scale details that Hooke could only dream of. We now know this vital cell boundary is just 7 nanometers thick, forming a totally fluid, moving mosaic. It's a lipid bilayer where molecules literally flip around, with embedded proteins acting as strict bouncers. Want to see those bouncers in action? Let's check out a super simple, classic potato experiment. Depending on the water's salt content, the potato's environment becomes isotonic, hypotonic, or hypertonic. The results? Pretty wild. A potato in plain water swells right up, but in salt water, it shrinks. Why? Because water desperately chases balance, crossing that membrane to even out the concentration. But here's a crucial question for you. Are human and plant cellular boundaries built exactly the same? Nope. Plant cells are neat, rigid boxes, while our cheek cells are kind of squishy and irregular. That's because plant cells have extra armor, a rigid cell wall, preventing a total structural collapse. Without that wall, animal cells are super vulnerable. In salty water, they lose water and shrink drastically. So I'll leave you with this puzzle. What happens to soaked mung beans placed into heavy salt water?